Osteoblasts are active bone-forming cells. These cells produce the bone matrix. They are the cells that form bone. In other words, they are the factories of bone that create new bone matrix. Its structure is completely different. It is typically cuboidal or columnar in shape, especially when they are active and forming bone. It has a round-shaped nucleus in the center. Due to the abundance of ribosomes, its cytoplasm is basophilic. Basophilic tissues or cells strongly absorb basic, that is, alkaline dyes. They appear blue or purple under the microscope. Inside, the rough endoplasmic reticulum and Golgi apparatus are well developed because these cells produce and secrete collagen type I and other bone proteins. They are arranged in a single layer on the outer and inner surfaces of the bone and perform four major functions. In the stage of formation, osteoblasts create the organic matrix known as osteoid, which is the structural unit of compact bone, and it is composed of collagen type I, glycosaminoglycans, and glycoproteins. After forming the osteoblast itself mineralizes this matrix. For this, it releases enzymes like alkaline phosphatase. This stores calcium phosphate crystals and converts the osteoid into hard bone. Regulation of bone remodeling. Bone remodeling is a continuous process in which old bone is broken down and new bone is formed, so that the bone remains strong, healthy, and updated. Osteoblasts produce two important proteins that keep the bone remodeling system in balance. The first is rankle. This protein is released by osteoblasts and stimulates osteoclasts. As a result, osteoclasts become active and start the resorption, that is, the breakdown of bone. The second is osteoprotogerin. It is the natural blocker of Rank-L. Osteoprotogerin inhibits osteoclasts. That means it reduces their activity so that bone resorption does not become excessive. In other words, osteoblasts not only build bone but also control the mechanism of bone breakdown. Hormone response 19 stimulates the Rankel system while osteoprotogerin counteracts it. Osteocytes are mature bone cells found in lacunae that maintain bone tissues and regulate bone remodeling through their long canalicular processes. These are the most abundant cells found in bone. That is, throughout the bone tissue, these cells are present in the greatest numbers. Osteocytes are the mature form of osteoblasts, but they are very different from osteoblasts in both structure and function. Osteocytes reside inside small cavities within the bone, which are called lacunae. Their structure is spider-shaped, with many long and thin cytoplasmic processes. These processes extend through microscopic channels called canaliculi, which directly connect each osteocyte to other osteocytes and to osteoblasts present on the bone surface. At the end of each process, there is a gap junction that allows ions, nutrients, and chemical signals to be transferred from one cell to another.